NMIT presents Lecture 7 on the topic of photosynthesis, the carbon reactions. This is part 1 of 3 parts of this lecture. This lecture is part of the plant physiology subject, which is a component of the agricultural and viticultural degrees offered at NMIT. Please refer to the recommended reading for Tays and Zeiger Chapter 8 for complementary information. Please ensure that you have watched the 3 parts from Lecture 6, topic Photosynthesis, the light reaction, before watching this lecture. My name is Dr. Nikki Cooley. This lecture will be separated into 3 parts. In the first part, we will be introducing the carbon reactions and the Calvin-Benson. We will be looking in some detail about the stages of the Calvin-Benson cycle: carboxylation, reduction and regeneration. In part 2, I will introduce C4 plants and the Kray's anatomy. This will be followed by an introduction and information on the crushalian acid metabolism known as CAM plants. We will end this lecture with part 3, which will look at the accumulation and partitioning of photosynthates, and we will learn about the formation of sugars, starch and glucose. It's always good to go over the basics again so that you can put your mind in the right place. The basics of photosynthesis is to turn the compounds carbon dioxide and water into sugars. We learnt that in the first lecture. The plants have sophisticated mechanisms to capture the energy from light and turn it into the chemical compounds. These compounds can be used by the plant. In the carbon cycle, we are going to learn about how this happens. If at any stage during part 1 of this lecture you are lost or confused, then please come back to this page to remind yourself of the objectives of this process. To continue our review, it is the capture of energy by plants which enables them to create these compounds or sugars. Solar energy is very important in this process. And to give you some idea of the dynamics, approximately 3 by 1021 joules per year is converted by plants to carbohydrates. This gives us approximately 2 by 1011 tons of carbon per year. Some of the subsequent products of the light reactions are the compounds ATP and NADPH, and these flow from the thylakoids to the stroma. The thylakoid membranes are where the light reaction occurs. They then flow into the stroma. They drive the reactions of CO2 and the production of carbohydrates. And it is this process that is the carbon reactions. Here we have an image of a chloroplast on the slide. The location of the light reaction, as we learnt in the previous lectures, was in the thylakoid membranes, and you can see these stacked in the image. The reaction of the Calvin cycle takes place in the surrounding liquid. This surrounding liquid medium is called the stroma. In the next few slides we're going to understand more about the Calvin-Benson cycle, but firstly I'm going to give you an overview of this cycle. If at any time you don't understand the detailed and complex chemical reactions, then please come back to this slide and review the simple concepts. This will help with the more complicated equations. Calvin-Benson cycle is a series of complex reactions that take part in the stroma in the chloroplast. This cycle is found in many photosynthetic prokaryotes and all photosynthetic eukaryotes. Carbon dioxide, that is CO2 from the atmosphere, is converted into compounds, and this process occurs during this Calvin-Benson cycle. The cycle's inputs include products from ATP and NADPH that are obtained from the light reaction of photosynthesis, and carbon dioxide from the atmosphere. Here are some important components of the Calvin-Benson cycle worth remembering. It is a cycle, and therefore it is able to regenerate its components. The cycle has 3 phases. The first phase is carbon fixation. The second phase is called reduction, and the third phase is regeneration. It is this third phase which enables the cycle to continue. We are now going to examine these three phases in more detail. So let's start with carboxylation. On your slide you'll see a molecule of CO2. This is currently in the atmosphere. This molecule of CO2 will absorb into the leaf. From there it will then absorb into the cell, and then it will absorb in the stroma of the chloroplast. So when the atmospheric carbon dioxide is in the stroma, it will enter the Calvin-Benson cycle and combine with a molecule called RUBP, or ribose 1,5-bisphosphate. This is a 5-carbon molecule, and this can be seen in the illustration on the slide. The grey balls represent a carbon molecule, and in this illustration you will see there are 1, 2, 3, 4, 5 carbons in this molecule. When RUBP is modelled, it looks something like this in real life, a very complex molecule. The process of binding the atmospheric carbon dioxide to RUBP is called carboxylation. It is also known as carbon fixation. Here we have another illustration showing the process of carboxylation. We have carbon dioxide and RUBP, and this combines to form a 6-carbon molecule. This 6-carbon molecule is very unstable, and so immediately breaks out into 2 3-carbon backbones or molecules. These 3-carbon molecules are called PGA, or phosphoglycerate. Each of these molecules of PGA also have a phosphate group attached. So, now what happens to the 3 PGA molecules? Well, they are what we call reduced. Let's go into this in some more detail. The 3 PGA combines with ATP to produce a molecule called BPGA, or biphosphate glycerate. This is a 3-carbon compound with 2 phosphate groups. You will notice that the ATP in this figure is illuminated with yellow, and this is to remind you that this ATP comes from the light reaction. The ADP molecule has changed colours, and this can remind you that it is ready to go back to the thylakoid membranes, where it can be converted again into ATP. Once PGA molecule has been produced, it then combines with NADPH. This is again a molecule from the light reaction. The NADPH separates both an electron and a hydrogen ion, leaving NADPH+. The process of the electron and the hydrogen ion allows a new compound to be produced called glyceraldehyde 3-phosphate, also known as PGAL. So to conclude, reduction is taking the molecule PGA and reducing it all the way down to this compound, glyceraldehyde 3-phosphate. This process involves both ATP and ADPH. Once the PGAL molecule is formed, it now leaves the cycle where it can combine with another PGAL molecule to form glucose. In the illustration on the slide, I have coloured in the carbon molecules. 3 are in blue and 3 are in red, each from the different PGAL molecules. You can see how they come together to form glucose. It is important to realise that PGAL molecules actually exit the Calvin-Benson cycle, and they go off to combine to produce glucose. So if one of these molecules exits, how does the pathway continue? At the beginning of this presentation, we were told that the Calvin-Benson cycle was in fact cyclic. So we need to look at how regeneration occurs. In order to understand how regeneration occurs, we need to understand about how many molecules are inputted into the cycle and how many molecules are exit. On the figure in front of you, we have a new illustration of the concepts that we have discussed previously. We are going to do what is now referred to as cycle accounting. That is, we count the number of compounds in the cycle and the number of compounds out. Let us start with the carbon dioxide, which combines with the RuBP to form PGA. This was the first part of the cycle and was called carbon fixation. In this cycle, there are 3 molecules of carbon dioxide that enter the cycle. These 3 molecules of carbon dioxide produce 6 molecules of PGA. There are a number of intermediate stages which we showed where ATP and NADPH are used in order to form the chemical structure of these molecules. As you can see, through the process of reduction, we end up with 6 molecules of G3P, and then this allows one of these molecules to exit the G3P, which goes on to make the sugar or the glucose, as discussed previously. But once you do the accounting, you realize that there are other molecules of G3P which can be used. In fact, there are a total of 5 of these molecules left. And these 5 are all reused or in the cycle, or regenerated. ATP, to be exact 3 molecules of ATP, are used to produce 3 molecules of RuBP, and this takes us back to the start of carboxylation. So we have 3 stages: carbon fixation, reduction, exiting of the 3GP, and regeneration, and hence the cycle continues. On the slide you will see a figure illustrating the Calvin-Benson cycle. This is the slide in the illustration used in the recommended textbook, which I will refer you to here. The process of the cycle that I have outlined to date is a simplified description of the cycle. There are concepts more complicated which you will not have to learn in this subject, but I will make you aware of so you can see both the complexity and understand that regeneration involves several intermediates. In this slide and the following slide, you will see the number of intermediates in the reaction. This table, 8.1, part 1, can be found in the textbook and shows 8 intermediates. This is the second part of table 8.1 and shows another 6 intermediates, giving a total of 12 intermediates in the regeneration cycle. The illustration on this slide demonstrates the chemical reactions of the intermediate steps within the regeneration stage of the cycle. As you can see, the complexity is impressive, and I will not be testing you in this subject to this level. I do expect you, however, to understand that 5 molecules of G3P are converted with 3 molecules of ATP back into RuBP, and this allows the start of the reaction. So thus, this enables the regeneration of the Calvin-Benson cycle. A very impressive cycle. There is one compound which is critical to the regeneration phase, and this is an enzyme called Rubisco. On the slide you will see a modelled image of Rubisco. It is an important component to the regeneration, and it has what's called a catalytic rate. That is, a rate that it can convert CO2. This catalytic rate is relatively slow. This describes the number of CO2 fixations per second. In plants, it is light that stimulates the activity of rubisco. In some plants, rubisco is inhibited by the presence of sugar. If the sugar is present at low levels, it may inhibit rubisco. High concentrations of rubisco are found in soya bean and bean. The inhibitor accumulates at the night and binds rubisco. It is removed in the morning by rubisco activase. In part 2 of the lecture on the carbon reactions, we will look at rubisco in a little bit more detail. So that brings us to the end of part 1, photosynthesis, the carbon cycle. This slide will summarise some of the points that I hope you learnt in this lecture. You will understand that there is a cycle that occurs in the stroma that allows plants to fix or capture atmospheric CO2 and convert it into sugars that are important for the plant's growth and development. You will understand that the Calvin-Benson cycle contains 3 stages. You will understand the important inputs and intermediates in these stages, as summarised in the slides previously. You should be able to account for the cycle. By this I mean be able to determine how many ATPs, NADPHs, and other molecules like RUBPH. You will understand that there is an enzyme called rubisco, and that this is important for the regeneration stage of the Calvin-Benson cycle. We will be going into more detail about rubisco in part 2. Some of the concepts studied and looked at in this lecture are quite complicated, so you may have to listen to this lecture several times before you understand. If you do not understand these concepts after you have listened to the lecture several times, then please raise this in the associated photosynthesis, the carbon cycle tutorial. For online students, this will be on a Monday, and for students taught in class, this will be the same week that we cover this subject area. Thank you very much for your time.